All right, thank you all for joining today. As a reminder, the public webinar series is designed to educate attendees on the scientific ecosystem within the Common Fund. And each month we'll have a representation from a data coordinating center and new developments from the centers that support them. So today I'm going to share a few scientific updates from the Knowledge Center, showcase some upcoming events, and then we'll hear from our featured speakers. So if you're unable to attend last month's webinar, I just want to remind you that the recording is available on our YouTube page here. And this webinar is being recorded and will be uploaded there as well. As I mentioned at the last session, we gave a brief update on the KC beta site. So this is the landing page and the link to it. In each webinar, as I said, we aim to provide updates on this funded program. So as the project manager, I wanted to provide some updates, and many of these are going to be undocumented mock-ups. And our latest one that I'm excited to share with you today is actually hot off the press. Coming to the gene page is the xRNA card, which you can see here, that allows users to examine RNA binding proteins related to the search gene using xRNA data. So the proteins, as you can see here in the red box, will be linked to their corresponding protein page. This is what it would look like on the protein page, and this is still a brand new page and is under development, but the xRNA card is the first content that will be available. So on this specific card, users can explore genes associated with the search protein. As you can see here, these will be also linked to the respective gene page as well. And then just as an added update, we're actively developing new cards for both Kids First and links. We'd love to see your own use cases and represent how you use these data from the various DCCs. So if you have any feedback, as this is a beta site, questions on how to use any of the pages or cards, or would like to be added to our listserv, please email us at help@cfdeknowledge.org. But before we get into our featured speakers, I just want to highlight some upcoming events that are happening within the ecosystem. We're thrilled to announce that our first CFDE exhibition booth and workshop at the Festival of Genomics, which is a genetics and genomics conference, will be held in Boston on June 12th and 13th. So come stop by booth 86, where we'll have interactive demos and educate attendees on the Knowledge Center, the Data Resource Center, and the various DCCs. We have been invited to host a workshop on June 13th, which is located in the Genome Dome, and this will feature the KC, the DRC, and 4D Nucleome. This conference is free, so if you're local and you have a moment to attend, we'd be happy to see you there and actually get to meet each other face to face. Later in the year, we'll attend an even larger conference in genetics and genomics, the American Society of Human Genetics. Our booth will also feature DCCs, including GTEx and hopefully Kids First, as we know both of these DCCs have attended in the past, and will also represent the centers and the CFDE. We are also going to be hosting an ancillary session, but we'll have more details to come as we plan further out. So be on the lookout for this as well. Other events within the ecosystem that I'd like to socialize include Bridge to AI Ready monthly speaker series in June, the Metabolomics Workbench virtual summer workshop in July, and IDG CFDE specialized courses in July as well. These details will be publicly available via these slides later after this webinar series, and it will also be on our Twitter if you need to look back. So today I'd like to welcome our featured speakers who are both from the Links Data Coordinating Center and the Data Resource Center. To give some background, Links is one of 14 resources within the Common Fund data ecosystem, and it serves as a data coordinating center. Avi Mayan will cover the Links datasets, tools, and a newly developed method to query the Links L1000 data for better prioritization of drugs, small molecules, and single gene perturbations. Giacomo Marino will then describe a specific tool that Avi's team has built to allow users to mine mammalian datasets across geo. But before I hand it over to our speakers, a reminder, please feel free to ask questions in the chat or at the end of their presentations. I'll be monitoring the chat. So with that, I will hand it over to Avi.
All right, thank you, Mackenzie. I'm gonna do screen share of portion of my screen. So I'm going to need a second. Get it right. Okay. So yeah, thank you so much for organizing this webinar series, and it's a great opportunity for us to showcase, our work. Today, I'm representing the Links Common Fund program, and also, as you may know, I'm also a PI of the newly developed CFDE Data Resource Center. So some part of this presentation will cover some of the activities that we are doing for this Data Resource Center for the CFDE. So the Links program was a common fund program that was for 10 years. It ended exactly 4 years ago. And we were the data coordination center for Links together with Stefan Schurer from the University of Miami and Mario Medvedovich from the University of Cincinnati. So a little bit about Links. So the concept of Links started with this, I guess, revolutionary seminal paper that came out from the Broad Institute, headed by Todd Golub, and the first author was Justin Lab. And I'm sure many of you heard about this concept of connectivity mapping. So the idea is to build a database where you have collected a massive amount of gene expression signatures for single drugs and other genetic or chemical perturbations. And then you can come in with your project, your biological interest. That can be a disease state versus normal, a mouse model, or some cells treated with some condition like a compound that you want to see what it does. And then you come into this links database, and then you can get mimickers and reverser signatures. So this was a paper that, it was a seminal publication from 2006. And then the idea for links was to really expand on this idea. So the links cube is this concept of having many, many perturbations. Again, those could be chemical, genetics, microenvironments. And then you apply those perturbations to different cell types. And typically those are cell lines because those are easily manipulated for high throughput. And then you measure what happens to the cells before and after the perturbation with different phenotypic assays that could be omics assays or imaging or other types of assays that measure changes to what happened to those cells. So in the second phase of links, so links had two phases. The first one was 4 years, the second one was 6 years. In the second phase, the program was expanded to have 6 data generation centers and one DCIC, which was us. And this is the map of where they were on the US map. And this is represented on the portal that we developed for links. As the data coordination center, and this will be a little bit relevant later, we are also a part of another common fund program called BD2K, Big Data to Knowledge. So we were serving on both sides. And the BD2K program was really about finding a solution to better manage data. And the CFDE is sort of like, I guess, a grandchild of that program. So one of the things that happened to us as the DCIC for links, we were awarded at the same time that those data generation centers were awarded, and we had some time that we needed to wait for the data to be generated. So during that time, we really decided that instead of waiting, we're gonna be able to just go to the public domain and create signatures from a lot of those publicly available resources like Geo and encode. And so basically what we've done, we wrote code scripts that takes this data and abstract it into signatures. And those could be gene sets. And those gene sets can be converted to what's now called knowledge graphs. So connecting the genes based on their shared attributes, or connecting the attributes based on their shared genes, as well as this concept of bipartite graphs. So you have the attributes on one side and the genes on the other side. So we've been doing that systematically across many resources. And this was a really successful effort. So one of the two bioinformatics tools that came out of this are Enricher and Harmonizome. So far, we process data from over 100 different resources. And Enricher currently has almost 500,000 annotated gene sets. And you can see here that there is now actually 72 million submission to the system. So this is a screenshot from a few weeks ago. And then over the last 10 years, these two websites together saw at least 2 million unique users. So those are really turned out to be very useful resources to the biomedical community, and many of you probably have heard about these. So one of the things that we've done for links was we had to build all those portals as the data coordination center. So we created the information portal, which is at the linksproject.org, as well as we had our own center portal, which is at linksdcic.org. And we also came up with this concept of an app store. So instead of building a portal that has everything in it, we really build a collection of tools. And this is not just our team. This is a collective effort of the entire consortium, and different groups develop those databases and tools, and we all serve them in this links tool marketplace that currently has 69 different tools, and all of those tools are represented as cards. So this is sort of like was a revolutionary thing for bioinformatics, having all those tools in one place, and here they're sorted by the number of clicks on those cards. So this is just another view of all of those cards with all those apps. And this was really, again, very successful. Our team, the DCIC, published over 45 bioinformatics tools and databases. We had over 2.2 million unique users. And right now, we still have about 4,000 unique users per day, 45,000 unique users per month. And also those tools, many of them have become very popular and successful with over 15,000 citations. So several years after the program started, the second phase of the program started, the Broad came out with this another seminal paper that was published in Cell. And here they introduced the Links L1000 assay and data. And this is the L1000 assay is a high throughput transcriptomics assay that is an alternative to microarrays and RNA-seq. And it measures 1,000 genes, but the rest of the genes are inferred relatively accurately. So you get the full genome, and they were able to do this transcriptomic assay in high throughput. And in the publication, they showed how they had 5,000 genes, loss of function or gain of function, some biologics, and about 20,000 small molecules that were used. And those small molecules were used in different concentrations across many cell lines. And also the gene expression was measured at different time points. So this was like a massive expansion of the original data set, and now it's even larger. So there is almost all of the genes were knocked out with CRISPR, and the small molecule collection reached 30,000, and the total number of samples that were profiled at the end of the program was 4 million. So one of the things that we've doing, some of those apps that we developed, and this is, I guess the L1000, you can say that this is the most substantial data set that was produced by Link, and also the most widely used resource that came out. So throughout the years, we developed search engines to query that data set, the L1000 signatures. So you come into those search engines with your data of, let's say, up and down genes, and then you get a ranked list of mimickers and reversers. And the most current app that we have for doing that is called SIGCOMM Links. It was published 2 years ago. And this is just a way that we were able to also make an impact by building those easily used search engines that were an alternative to what was provided by the Broad. And here is just one example of how those search engines have been used. So far, for those 4 tools that I just showed you, they were cited by about 800 papers, but many of those papers really are experimental validation of predicted drugs from those hypotheses that could be generated from those search engines. So this is one of the, I guess, one of the most successful implementations of this approach with our tool. And this project we were actually part of. And this was a collaboration with Chiara Giannarelli, that is now a professor at NYU. So Chiara, her idea was like to take PBMCs and then treat them with plasma of patients that have atherosclerosis, which is the clogging of the arteries. So in the plasma of those cells, there's some signals that promote the formation of foam cells. And what we wanted to find is small molecules that can potentially reverse that signature. And then one of the drugs that came out from this analysis, one of the reversers that came out from the querying of the RNA gene expression signatures from those stimulated PBMCs was seracatinib, which is an AstraZeneca drug that in cell-based assays, animal models, and specifically a rabbit model of atherosclerosis, showed like a significant reduction in plaque formation. So this project almost came all the way from concept to clinical trials. AstraZeneca decided that they do not want to proceed with clinical trials for this phenotype, for this drug, for some reasons, but we almost made it all the way through. And that was an exciting project that was published very recently, last year, in Nature Cardiovascular Research. So this is the SIGCOMM Links interface, where you come in with your up and down genes, and then you press submit, and then you get those ranked list of drugs. If you don't have up and down genes, you can start with a single gene or a search term like a disease, and then there's different ways that you can convert a search term into an input that then you can submit to SIGCOMM Links for getting those prioritized compounds. So the program has different sources of inputs, either a single gene, a gene set, up and down genes sets, or any search term. So the topic of this presentation today is really about a new method to query the Links L1000. So if you think about it, there are about 1 million gene expression signatures in the database that we query. And when you get the result could be overwhelming. And then there is this issue of having the very specific signatures for specific drugs in a specific condition. And one of the things that we are thinking is that we can create some type of a consensus approach to rank those drugs. So for example, if we have this drug, valdecoxib, many times in the returned results. So let's say if we're searching for compounds, there are 700,000, a little over 700,000 entries in the database. And then the drug in different cell types, conditions, concentrations is showing up in the results in many places over the ranks. So what we want to see is the ranks of that drug concentrated at the beginning? So you can do it by looking at the ranks of the drug, or we can look at the score, whether it's a reverser or a mimicker, and what is the p-value or the z-score of the ranks. So we can compare that deviation from the expected rank or from the expected collection of scores, and then can prioritize the drugs now at the drug level, not at a specific condition. So just if I'm showing you here what the results right now in SIGCOMLIX look like, there is the drug name and then the cell types, the concentration, the batch, where it came from, and then the time point. So this is going to be potentially replaced by this consensus approach, or alternative to this, you can have this consensus approach that finds the AUC of the drug across all of the instances of it appearing in the results. And then there's going to be a p-value that is computed by the KS test and all of the conditions that were identified for that drug. And recently we applied this approach to a collaboration with two investigators from the University of Wisconsin, Mark Keller and Alan Atty, who are studying diabetes. And they're using this diversity outbred mice as a model for human disease. So there's mice, they were collected from different parts of the world to create this collection of genetically diverse mice that now you can do all kind of like creative experiments with. And they have 500 of those mice that are genetically diverse. And what they've done, they took out from those mice islets, and then they want to see how those islets are, how well those islets produce insulin. So they have applied different assays, first to profile the islets of their ability to produce insulin, as well as they did RNA sequencing of those islets. So for each mice, they have a sample of RNA-seq from those 500 diverse mice. And then what you can do, you can look at the insulin secretion. So this is a visualization of the insulin secretion for across the mice. So the mice are the columns, and the level of insulin secretions are the rows. And then you can see that there is a range of for different mice based on their genetic background. They're more or less responsive to secreting insulin. And then you can take the RNA-seq data, and then you can see how individual genes based on their expression correlate with insulin secretion. So now instead of having like differential expression signatures, we have genes that are highly positively or negatively correlated with insulin secretion. And this is just showing that this gene E4B4 B4 is highly correlated with insulin secretion. So at least for many of those known genes, you can see that they come in either on those edges of this ranking based on this correlation. And then what you can do, you can submit it to SIGCOMLIX, and we use this new approach, this consensus signatures KS test approach. And to test those drugs, they also needed a positive control. So this is a drug called ISX9 that is known to when you treat the islets with this drug, they will produce insulin. And then this is the results of the screen. So they tested about 30 drugs, and you can see that we're really enriching for compounds, and there's several compounds that are doing much better than our control compound in mouse islets. And then the next step is now they're testing it in animal models as well as in human islets that are isolated to see if this also translates to human studies. So now I'm going to switch gears a little bit, and I'm going to talk about the CFDE. And the history of CFDE, like I mentioned before, it started with BD2K, and then there was this program called the Data Common Pilot Phase Consortium, the CPPC, and then the CFDE came after that. And then throughout that time, we were thinking a lot about finding solutions to data management, data analysis, and how to do those bioinformatics analysis in a better way. And we have this sort of like timeline of publishing those different tools that sort of like have some type of succession, and were developed as we improved our understanding of how to solve those issues. So in 2016, we published this paper that is actually a Jupyter notebook. And this is in this journal called F1000 Research. And this is the RNA-seq pipeline that did some analysis on Zika virus. And the idea was that the paper is coming out with a Jupyter notebook that contained the paper in it. And then readers of the paper can take that paper and swap the input data and have their own RNA-seq pipeline analysis with all of the figures and results based on their work, on their signatures. So this paper sort of got some attention, and then several years later, there was a successor to this paper called BioJupys. And BioJupys really took advantage of this concept of Jupyter notebooks and some of those Jupyter notebooks libraries that can automatically execute the notebook. So with BioJupys, you can come in with your RNA-seq data, you upload it. It can be FASTQ files, or it can be a data matrix with your data. Then you have to make some choices of the tools that you want. You have to select your perturbations and control samples. And then you press submit to get a report that looks almost like a paper with your RNA-seq analysis. And that tool is still widely used by the community, and it's been a really great success. But then Daniel Clark, who joined the lab at that time, thought of even a better way to sort of like generalize this. And this when Apiters came. So Apiters was a successor of BioJupys, and Apiters is not really a bioinformatics tool. It's sort of like a way to develop tools in a more sustainable and easy way. So many of those Apiters can replace some of those bioinformatics tools that I showed you that we developed for links. And the way Apiters work, you start in with a Jupyter notebook, and then you annotate your notebook in a way that you can convert the notebook into a full stack web-based application that has like a input form. You upload your data, you set up some parameters, and then you execute the notebook in the cloud by pressing submit. And then you get those reports that again look like publications. So far we published over 100 of those Apiters, and all of those are served on this Apiters catalog, and they're still widely used. But then the next thing that we've done was to develop this sort of like the next step from Apiter, or the successor of Apiter, was this concept of the Playbook Workflow Builder. And this is a CFDE partnership that, again, led by Daniel's work on coming up with this idea of taking the APIs from the various Common Fund programs, as well as many other sources and tools, and creating this network of microservices that can be chained to build workflows. So this is the graphical user interface workflow, bioinformatics workflow engine that I would like to quickly demo. So let me switch. So this is the website, and I will post the URL shortly. And here you can come in and you see all those cards, and those cards tell you what you can start with. So for example, you can start with a gene. And then here I can type in STAT3, for example, which is like one of my favorite genes. And then I'm presenting with another set of cards of what can I do with this gene. And those are different next steps. And you can see the breadcrumbs at the top show me how I'm expanding things. So here I can click on this query GTEx for median tissue expression. And immediately I'm getting results. So this is actually happened in real time, where it went to the GTEx portal and it got the expression of that gene across tissues, and then press extend. And now I can visualize this, for example, as a bar plot. And this takes a minute, but here you get a bar plot with the tissue expression of the gene across the tissues. And then what you can do, you can switch here to report mode. And the report mode actually wrote a story of what happened. So we started by selecting STAT3, and then we queried against GTEx, and then we visualize it with a bar plot. And this is a report that was generated, and then we can export the report, and then we can change the workflow. So we can go back to the workflow mode, and we can continue to explore things, or we can start again with another gene and change the gene and get that result. So this is still a work in progress. Let's switch back to our presentation. So overall, like as we are proceeding with working on the CFDE data resources, there are several apps that are emerging. We haven't stopped collecting signatures and converting resources into signatures, and we had this double drop of RamaGene and RamaGeo resources that you will hear about from Giacomo. And then everything is going to be served from the CFDE workbench, which also I can share the link from. And then there's some other apps that are sort of like in the works. So before I pass it to Giacomo to talk about RamaGeo, I would like to first thank the members of the lab. Daniel has been instrumental for working on the second part of the presentation, and Errol is also working on the links signatures part that I mentioned. And also you can see that we have a lot more faces on this picture plot, and this is because we have a summer program every year where we double the size of the lab, hosting 10 to 12 summer undergraduates, and they are starting on Monday again this year. I also like to acknowledge the CFDE team for their great support, and then Shankar for collaboration, and his team for collaboration on the DRC, as well as our collaborator at Wisconsin and NYU for those experimental projects that I mentioned, and also for the future collaborations with the Knowledge Center, and for organizing this seminar, as well as the partners, the Playbook Workflow Builder partnership that includes other members of the CFDE, as well as our membership in the Data Distillery Knowledge Graph partnership, which I did not talk about today. So I will stop sharing.
Questions for Avi before we move on? Here's one. from Henning. The playbook looks very user-friendly. What is the reason not to use one of the well-established workflow management systems?
So it's not exactly the same as the workflow management systems, but it is compatible with CWL. So one of the things that the playbook has is it has a lot more details about each step. And it also has this network where you can see what other parts are available at each step. So current workflow engines don't have this visualization of what you can do at each step as you expand the workflow. Another thing that we're working on that I didn't mention is that we are using LLMs to construct those workflows automatically based on prompt or through chat. So the idea is that you'll prompt or you'll say what workflow you want, and then we will search the network to see what is the most fit workflow that can be generated based on what you want. We may ask you questions along the way, and then you will get the workflow immediately. And this is only possible if you set it up this way, where you have this network of connected components that are only connected to the things that are possible. I forgot to mention that in all of those 6 apps that I mentioned in the last slide, we are integrating different parts of those LLMs to enhance the functionality, and we use those LLMs for various things. And Giacomo is going to touch on that a little bit in his presentation.
Beautiful. Henning says thanks. All right, Giacomo, over to you. Again, folks, if you have residual questions, please post them in the chat and or the Q&A, and we can answer them for you as we go.
Right. So hello, everyone. My name is Giacomo Marino, and I'm a bioinformatics software engineer in the Mayan Lab, the Icahn School of Medicine at Mount Sinai. I'm a member of the DRC team in the Lynx DCC. And today I'll be speaking about one of our new resources called Rumageo. So just to begin with an overview of the project, the main goal of this project was to automatically compute signatures from uniformly aligned bulk RNA-seq studies extracted by Geo from our resource ArchIS4. And we do this through identifying conditions using metadata string embeddings and then k-means clustering. We can also sort of verify our samples partitioning at the data level using a silhouette score. And additionally, we can extract functional terms from the geosample and study metadata, as well as associated abstracts and summaries. And this we can use to help summarize gene set search results. To serve all of these gene sets, because it's quite a large collection, we utilize and adapted the framework that was built for Rumagene, which has automatically extracted signatures from PubMed Central articles, which was developed by Daniel Clark. And this allows us to serve those gene sets for enrichment analysis, PubMed search, as well as metadata search. So this is an overview of the workflow of Rumageo. So we start with all of these aligned, uniformly aligned human and mouse studies. And then we have some constraints on the number of samples, as well as filtering out samples that are likely from single cell experiments. And then, like I said, before we use these sample metadata string embeddings of some of the key sample attributes, as well as k-means clustering to partition those samples into various conditions. And we can create titles that describe those conditions by looking at shared words across those group samples. After this, we can look at, and see if we find any keywords that would denote a normal condition, such as wild type, control, and then use Lima-Boom to actually compute differential gene expression signatures and extract up and down gene sets based on the adjusted p-values. We'll basically just discard sets where there weren't enough significantly differentially expressed genes, so less than 5. And then for those that are greater than 2,000, actually incrementally lower the threshold to get below that number. So this is an example of this metadata attribute embedding that I was speaking of. So on the top row here, we have the metadata string embeddings as a PCA for this for the study that was looking at gallbladder, tumor, and normal tissue from 4 different patients. We can see in the top, we achieve a pretty nice separation of the tumor and normal. And what's differentiating them here really is just the patient number that we see in this metadata. Whereas in the data level, we see for 2 of the patients, it looks like the tumor and normal are grouping together quite well. So this may speak to the data's quality, but this is part of the reason why we chose to sort of use the metadata to actually partition and group the samples. And then we can actually evaluate how well that aligns with the data afterwards, which is done with this data level confidence score with this silhouette coefficient. And this basically reflects the tightness and separation of the clusters with a value of 1 to -1. And we can see here on the right, this is the distribution of silhouette coefficients across human and mouse. And we see this sort of bimodal distribution where sort of a lot of the studies cluster quite well around that 0.5 or above, and then some cluster not quite as well or don't have as much agreement at the data level around 0. Looking at sort of the contents of the RomaGeo database, we can sort of observe a few different things. One being that this first row or column here, we have human on the top and mouse on the bottom. This is looking at the genes and then how many gene sets they are contained within. So we see a majority of genes are contained in very few gene sets, and a few are contained in many gene sets, like up to around 10,000. We see in the figure there. And then we have a distribution of the gene set lengths, with the y-axis is log scaled. So there's sort of a lot of smaller gene sets and then sort of an equal distribution moving outward towards 2,000. And then another sort of thing we can look at is how many gene sets are we getting per study. And we can see this periodic nature of this chart. And that's sort of arises from the fact that a certain amount of conditions, if you do some combinations, can lead to a certain amount of gene sets. So for instance, 3 choose 2 or 4 choose 2, and that's what's leading to these sort of peaks. We can also look at the groups per study, and here we see sort of something interesting in the fact that there is sort of a bias towards an even number of groups. You can see 3 and 4 being equal and 5 and 6, but the majority being sort of 2 experimental groups there that are being compared. And we can also look at the number of studies per year, and this was taken from sort of a mid-year 2023 release of ArchES4, and we're currently updating the resource. So you could see there's some drop off there. And then looking at the actual metadata from these studies included in the RomaGeo database, we can identify a variety of functional terms, including tissues and cell types, cell lines, diseases, and genes. And we can look at both how many sort of unique occurrences we have of those, as well as the total amount contained within studies. So here we can see really the most common functional term that we're pulling out are definitely the tissue and cell types, but many other functional terms can also be extracted. And from those genes, we can also look for kinases and transcription factors, which we'll use for benchmarking later on. And we can see that we have quite a sort of comprehensive view. Those transcription factors, about 550 unique across for both human and mouse, and about 200 kinases for human and mouse as well. Visualizing all of these gene sets for both human and mouse, we can see that there is sort of a fair amount of separation for human and mouse, with some sort of density of where they overlap here at the top. So this is looking at sort of harmonized gene symbols between both human and mouse here. And then we can also color this UMAP by various metadata attributes that we previously identified. So on the left here is by the 10 most common tissues. And we can sort of visually observe that we see some coherency in clusters of these tissues here that we see from the signatures. And then on the right, we have the 10 most common diseases that were identified. And again, maybe it's slightly less coherent, but we definitely see some clusters of these various diseases that we identified in the geo metadata. Another thing that we can do is look at where RamaGeo gene sets fall in the enricher gene set space. So on the left is just the enricher gene set space. And then on the right, we overlay all of the RamaGeo gene sets on top of that. And we can see that most of this overlap is in this area here, which is actually corresponding to the enricher crowd category, which has a lot of user submitted geosignatures. So this makes a lot of sense. And additionally, we can color this by transcription factors and kinases that we saw before. So this is a little bit more sparse, but we can sort of observe some clusters of these transcription factors and various kinases here on the UMAP as well. So with those transcription factor gene sets, we know that they're sort of in there. They're in the signatures. So one approach that we tried was to create a correlation matrix of coding genes from the randomly selected RamaGeo signatures. And then we take the top 200 correlated genes with each transcription factor. And this allows us to create a transcription factor gene set library from RamaGeo. And we can benchmark this with a variety of libraries. And we see that the recovery is actually quite good for transcription factors in that it's ranking the correct transcription factor very highly across many of these benchmarking libraries. And we can use a similar procedure to create a kinase gene set library from RamaGeo. And though this doesn't perform quite as well as the transcription factors, we can still see better than random recovery of those kinases. Another sort of interesting analysis that we conducted was to take the human, in this case, RamaGeo gene set library, and transpose it. So now we're looking at genes instead of gene sets. And if we cluster this collection of genes and what gene sets they're in, then we find these, in the case of humans, 77 functional groups. And we can then look at the functions of these clusters from a select set of enricher libraries and see if we're seeing consistent, significant enrichments. And we can see for a majority, about over half in the humans case, we have these sort of quite clear functions for what those clusters correspond to. And then some of the unlabeled clusters, we can see sort of correspond, many of those genes are from the same chromosome, or perhaps they have sort of multiple functions, so it's not quite as clear. But this is a sort of an interesting analysis. And by looking at these clusters, we can also perhaps illuminate some knowledge about understudied genes as well. And so this is the same analysis done for the mouse collection of genes. And in this case, there were 67 functional groups. And for those, about 2/3 had pretty clear and consistent enrichments for a certain function. So just moving on to the actual site now, it currently contains 135,000 and 158,000 human and mouse gene sets, respectively, from over 23,000 geo studies. And we have sort of three main methods that you can actually query this data, which are the gene set search, PubMed search, and a metadata search. And this is all currently available at ramageo.com, which so feel free to explore the site afterwards. After you submit a gene set, we'll automatically detect the organism. So that's either human or mouse, depending on the gene symbol. So there's separate backgrounds for those. And then you'll be served with these matching signatures with the enrichment statistics. Clicking on any of the condition titles will show you actually the metadata attributes and the samples corresponding to that condition. And additionally, you can click on the overlap or the gene set size to look at the actual genes. You can copy those to the clipboard or submit them to Enricher or back to RamaGeo as well. Sort of another thing that we did was looked at these functional terms that we could extract from PubMed abstracts associated with these studies or from the geo summaries themselves. And then we sorted those into categories using an LLM. And these are 4 categories. So we have disease phenotype, biomolecules, tissue/cell type, and pathway or biological process. And here we can actually look at the enrichment of those terms in unique studies that were enriched with your inputted gene set. So you can get a sense of the overall sort of what are those studies entailing that were enriched. And we'll show a bar chart that visualizes the negative log 10 of the p-value. Additionally, below that bar chart, we can see the actual terms and then the counts that lead to that p-value for the Fisher's test, as well as you can click any of those terms, and then you'll be shown a list of all the gene sets that corresponded to those that led to that term enrichment. And below that, we also have just a word cloud to help visualize those terms as well. The last sort of tab on these gene set search page is concerns enricher terms. So we pre-compute significantly enriched terms from selected libraries, including CHIA, KEGG, WikiPathways, MGI, GoBP, GWAS, and the Human Phenotype Ontology. And then we can look where these significant enricher terms rank among the top 500 signatures. And we can compute the significance of that with the KS test. So this helps show what are those actual highly ranked gene sets being enriched for generally. And then speaking sort of to what Avi had alluded to with the integration of LLMs, one thing that we integrated here on Rumageo was this hypothesis generation module. So here you can actually click this little hypothesis button when you do the gene set search, and you'll be prompted to input a textual description of the gene set you submitted. And then it will take that description as well as the description of the geostudy, and then enrich terms from the overlapping genes from those two gene sets to create some hypothesis about why that overlap may exist. You can also filter this table by a search term and download all the results as a TSV. And then for PubMed search, you can search for the PubMed API and then get returned with papers that have corresponding gene sets in Rumageo. So you can expand that on any of these after you search. And then we also have a metadata search to directly search that geo metadata, and then we'll sort of bold the search term in the results. So you can grab maybe specific gene sets you're interested or signatures you're interested in. We also have all these gene sets available for download, as well as the co-occurrence matrices that I referred to earlier. There's also a user manual help page where all of the functions of the site are explained. And in the future, we will like to add up and down gene set signature search, since we have these paired signatures. We'd also like to auto update the site as new studies are aligned for ARCHES 4. Certainly, there's a lot we can do to expand this gene-centric analysis that I showed before, and perhaps also serve interactive view maps on the results page, and also add better ways to prioritize or more ways to prioritize and sort the results of a gene set search. And finally, we can also cross Rumageo and Rumagene to discover unexpected associations between studies. And with that, I'd like to just thank everyone in the Mayan lab, in particular Daniel and Eden, who worked a lot on this project, and also thank you all for listening. So that's it for me.
Thank you so much. Does anyone have any questions for Giacomo? All right. Well, thank you so much for your presentation today. Before we leave, just want to say if you joined us late, the slides will be available and the recording will be available to all on our YouTube page. Just one last reminder, our next event will feature an overview of the Illuminating the Druggable Genome program, which will be our next session, Friday, January 28th. So we hope to see you then. Thank you all so much for your time, and we hope to see you at our next session. Bye.
Thank you.